Hello fungi and welcome to the first channel about fungi. Today we will talk about composition of fungal cell wall. Similarly to plants, cells of fungi are surrounded by a rigid construction, called cell wall. Cell wall is located right above the cellular membrane, and it protects cells against environmental factors such as osmotic stress, UV radiation, oxidative stress. Cell wall also determines the shape of the fungal cell and has a great role in metabolism and exchange of ions, interaction with another fungal individuals, as well as host defense mechanisms. In general, cell walls of fungi have two principal parts, a relatively conservative inner part of the construction and a variable outer part. Conservative part usually consists of layer of polysaccharides like chitin and beta-glucans, branched beta-1,6-glucan and linear beta-1,3-glucan. These polysaccharides bound to each other, making mesh-like structure around the cell and serve as a backbone for attachment of another components of the cell wall. At the same time, variable outer layers have a species-specific composition which directly affects wall function and is tailored to the physiology of fungus under specific conditions. Outer layers are highly diverse and probably there is no other structure of the fungal cell that exhibits more phenotypic diversity than the cell wall. Now let's talk briefly about the composition of cell wall of the main human pathogen filamentous fungus Aspergillus fumigatus. Aspergillus fumigatus is one of the main pathogenic fungi of humans. Daily, we inhale thousands of canidia of this fungus. Immune system of healthy people easily eliminate canidia from the body, but people with impaired immune system are not able to combat pathogenic fungus. Canidia disseminate in the body and fungus switches to filamentous growth, which cause acute aspergillosis, which may have lethal outcome. So cell wall composition may change during a specific stage of the fungal life cycle. For example, canidia and hyphal forms of Aspergillus fumigatus have slightly different components of the variable part of the cell wall, which helps perfectly exploit fungal adaptation and pathogenicity at every step of fungal stage of development. Here we see a conservative part of the cell wall, but the fact is that the chitin and beta-glucans are highly recognizable by host cells as pathogen-associated molecular patterns, and the fungal cells are attacked and removed by host immune system. In Aspergillus fumigatus, both canidia and hyphae, cell wall contains alpha-1,3-glucan. This polysaccharide is located right on top of the conservative part, contributes to pathogenesis by concealing the pumps from detection by the immune system. Also, in Aspergillus fumigatus, alpha-1,3-glucan is essential for aggregation between swollen canidia. Next layer of canidia cell wall is melanin. Melanins are the family of pigments due to which canidia of Aspergillus colored in dark green. Melanin contributes to the ability of fungi to survive in harsh environments, including photoprotection and thermal regulation. In general, melanin significantly enhances the virulence of many important human pathogenic fungi. And the upper layer is hydrophobin, highly hydrophobic proteins that form rod-like structures on the canidia surface. Sometimes in the scientific literature, hydrophobin is called rodlet protein. The name of this compound speaks for itself. It makes canidia hydrophobic, so fungal spores remain dry in any conditions, which support their better distribution. During germination, the cell wall of canidia undergoes changes, and cell wall of hyphae has slightly different composition. As I said before, polysaccharide alpha-1,3-glucan covers conservative part of cell wall, making chitin and glucans not visible for immune system of fungal host. There are two main components located on the top of alpha-1,3-glucan. Galactomannan is a polysaccharide crucial for vegetative and polarized fungal growth. Being a unique component of Aspergillus cell wall, detection of galactomannan in blood and body fluids of humans is diagnostic sign of invasive aspergillosis. And the last, polysaccharide galactosaminogalactan is the upper layer of hyphal cell wall. It's highly important for protection of hyphae during active growth, and also it takes part in biofilm formation, establishing an extracellular matrix between hyphae. Galactosaminogalactan is expressed during chronic and invasive infections, and its production correlates with the intrinsic virulence of Aspergillus species. As you can see, pigment melanin is not present in the hyphal cell wall. That's why hyphae of Aspergillus fumigatus lose dark green color that the canidia were endowed with.